Before opening a discussion that has been tearing apart the modern scientific community for approximately 40 years until today, and that does not seem to be about to cease, it is probably worth considering what a staminal cell is. A widely accepted definition of a staminal cell is that of a cell having two fundamental characteristics: the capacity of unlimited or extended auto-renovation, or in other terms, of reproducing multiple times without undergoing the differentiation process, and the capacity of generating intermediate progenitor cells with unlimited reproductive potential, from which populations of highly specialized cells descend. In other terms, stem cells can reproduce themselves indefinitely until a particular genetic signal, still being investigated, transduced into a chemical signal, triggers the cells to undergo a sort of metamorphosis, through which they become highly specialized cells. It is necessary to remember that there are many different kinds of cells in the human body, each characterized by a unique shape and set of functions. Considering their huge regenerative potential, the possible applications of staminal cells to the medical field are clearly endless, varying from cancer to diabetes, from the Parkinson syndrome to blindness, or also from cystic fibrosis to lesions of the spinal cord, just to make some examples. But where do stem cells come from? Is it completely safe to operate on humans with this sort of technology? Is it fair to sacrifice a human life, even if it just began, to save a person in need? These are the kinds of questions that have been posed multiple times in the past decades and have slowed down, on a certain extent, the applications of such small yet precious cells to therapy. From a strictly scientific point of view, the preparation of human embryonic stem cells implies the production of human embryos or the usage of excess embryos that result from in vitro fertilization, the development of such embryos up to the initial blastocyst stage, approximately 150 cells, and the withdrawal of the cells from the embryoblasts or inner cell mass, ICM, an operation that implies the destruction of the embryo. Taking stem cells from a human embryo indeed does kill the embryo itself. There is no way back. An embryo deprived of its stem cells is blocked even before starting. The cells are then cultured on a lawn of fibroblasts and proper media, where they reproduce, giving rise to colonies known as embryoid bodies, which in turn are grown until they generate cellular lines capable of reproducing indefinitely, while maintaining the characteristics typical of staminal cells for months or even years. However, the usage of stem cells is not entirely safe. Mostly, yes, but not entirely. The cells, first of all, could be rejected by the host organism, which would force the patient to be treated with immunosuppressive drugs associated with many unpleasant side effects for the rest of his life. And secondly, the inserted cells could not be regulated properly, in that they have been inserted in a foreign body and started to grow uncontrollably, which could lead to the formation of tumors. Now, leaving the side of science for one moment, based on a correct and complete biological analysis, starting from the fusion of gametes, the living human embryo is a human subject with a well-defined identity, who at that point begins its own coordinated, continuous and gradual development, in such a way that it cannot be considered as a mere aggregate of cells in none of its successive developmental stages. Being a human individual, an embryo is entitled to its own life, which means that every intervention that is not to favor the growth and development of the embryo itself represents a detrimental act towards that right. Consequently, the withdrawal of the inner cell mass of the blastocyst that damages the human embryo gravely and irretrievably, interrupting its development, is a solemnly immoral act comparable to murder. There is no such thing as a purpose considered to be good, one of the many therapeutic advantages conferred by the adoption of stem cells that can justify an intervention like this. A good purpose doesn't make right an action which in itself is wrong.
There are now many examples of some adult stem cells that, just like an embryonic stem cell, have the flexibility to form any tissue in the body. There are at least 72 different diseases where human patients have improved after treatment, and that includes things like leukemias and anemias, including sickle cell anemia. thalassemias, heart damage, chronic liver damage, stroke damage, growing new corneas to restore sight to blind patients, and the list goes on and on. In the meantime, there are no treatments for humans with embryonic stem cells, and in fact, most of the animals die in the embryonic stem cell experiment rather than being helped.
An interesting solution to this scientific and moral debate, indeed, appeared when pluripotent staminal cells were discovered in diverse human tissues, such as in the bone marrow, in the brain, and in the blood of the umbilical cord. And above all, when thanks to the significant contribution of the Nobel Prize-winning stem cell researcher Shinya Yamanaka, the possibility of reversing the biological clock of specialized cells by turning already differentiated cells into fully pluripotent non-differentiated cells called induced pluripotent stem cells, and then driving their development in whichever direction, resorting to proper genetic signals and media, became an option. With this approach, adult stem cells, previously pushed into the background in that they were thought to be capable of differentiating only into a limited number of different types of tissue, acquire a major role in finding a compromise between the therapeutic ambitions stimulated by the usage of such available source nature itself provides, and the great sense of responsibility required while dealing with the dignity of every human being.